Welcome to This Explainer. Today, we're jumping straight into a truly fascinating piece of scientific literature that honestly directly impacts what might be on your dinner plate tonight. We're breaking down a master's thesis titled Genetic and Phenotypic Differentiation of Nile Tilapia Revealed by Molecular Markers. It was authored by Dr. Ahmed Dorgham, a scientist at the Fish Breeding and Genetic Department within Egypt's Central Laboratory for Aquaculture Research. And his work? Well, it uncovers the hidden science, the molecular secrets, and the physical traits of literally one of the most vital food sources on our planet.
So let's just dive right into the core of this, because understanding the genetics of the Nile tilapia is absolutely crucial for the future of our global food supply. According to the UN's Food and Agriculture Organization, tilapia is the second most cultivated fish in the entire world. It's only beaten by carp. It's farmed in almost 100 different countries. But here's the catch. To keep feeding a rapidly growing global population, we have to make sure this fish remains resilient, healthy, and capable of fast growth. And to do exactly that, scientists like Dr. Dorgham are looking deep inside its DNA.
To give you an idea of the sheer massive scale we're talking about here, the Nile tilapia represents a highly domesticated livestock industry with global sales blowing past $2 billion a year. It is an absolute economic powerhouse. In Egypt alone, tilapia makes up nearly 70% of their entire fish culture production, which actually makes Egypt the producer of over 20% of the world's farmed tilapia. So we're not just talking about some niche theoretical science topic. This is a multi-billion-dollar pillar of global food security.
But to really understand where we are today, we have to trace back its incredible history. The farming of tilapia is actually believed to have started over 4,000 years ago in ancient Egypt. Fast forward a few millennia, and the first scientifically recorded culture of the fish took place in Kenya in 1924. However, it wasn't really until the 1960s and 70s that the Nile tilapia just completely exploded onto the global scene. Thanks to its crazy adaptability, excellent growth rates, and tough resistance to disease, it was shipped all over the world as this sort of miracle food source.
Now, here's where things start to get a little dicey, and it's a classic biological bottleneck. The vast majority of worldwide Nile tilapia production today is actually based on just 6 initial sources. We're talking specifically about stocks collected from places like the Ivory Coast, Ghana, Egypt, and Uganda. These founding stocks were established from a painfully limited number of wild fish decades ago. When you take a highly prolific fish that breeds incredibly fast, and you literally build an entire global industry from a tiny handful of ancestors, you're going to run into a very specific genetic danger.
And that brings us to our hidden villain for today, inbreeding depression. Because tilapia reproduce so quickly from such a tiny original genetic pool, they are incredibly vulnerable to this phenomenon. See, when commercial farms simply take fish from their own ponds, or, other limited commercial sources generation after generation, the genetic variability just absolutely plummets. So the bottom line here is that without genetic diversity, we risk losing the exact traits that make tilapia such an amazing, adaptable food source in the first place. A diverse genetic stock naturally gives you the traits farmers and consumers are looking for: fast growth, high survival rates, strong resistance to diseases. An inbred stock, on the flip side, suffers some pretty detrimental effects. They grow slower, they're physically weaker, and their commercial viability completely drops. It's essentially a ticking biological time bomb if it isn't strictly monitored.
To tackle this, Dr. Dorgham's research kicks off with something called morphometrics. Morphometrics is basically the science of judging these different strains of fish by their physical cover. It's the quantitative analysis of size and shape. Before even looking at the invisible DNA, the researchers wanted to see if they could actually differentiate between a totally wild tilapia population collected from the Ismailia Canal and 2 farmed, commercial populations from Kafr El-Sheikh and Fayioum simply by measuring their physical bodies.
Researchers meticulously measure the physical traits of these fish, completely transforming physical shapes into hard, analyzable data. They aren't just tossing the fish on a scale. They're taking highly precise measurements of body length, head depth, the distance between the head and the pectoral fin, tail thickness, the whole shebang. And then, every single one of those measurements is divided by the standard length of the fish. That way the data isn't skewed just because one fish happens to be older or overall larger than another.
Using a really advanced statistical sorting tool, specifically stepwise discriminant function analysis, they use these physical markers to actually categorize the fish into distinct populations. And what they found is that out of all those measurements, certain key traits were incredibly predictive: harvest weight, head length, body depth, and that distance between the head and the pectoral fin were the major defining characteristics. Literally, just by analyzing these numbers, the statistical model could correctly classify a fish's origin with mind-blowing accuracy.
And when we look at the physical growth results after an 84-day feeding trial, the farmed Kafr El-Sheikh tilapia is the undisputed heavyweight champion. They reached a final mean weight of over 50 grams, growing significantly faster with a daily weight gain of roughly 0.53 grams a day. Meanwhile, the other farm strain from Fayioum and the completely wild Abbassa strain really lagged behind, maxing out at roughly 42 grams. So on the surface, it definitely looks like the Kafr El-Sheikh farm is doing everything right? They're producing a much meatier fish.
But physical size isn't the whole story. So let's zoom way into the microscopic level and see how this builds our understanding of their underlying genetic health. Just because a fish is bred to grow fast, it doesn't mean its genetic foundation is actually secure. To really see what years of domestication and selective breeding have done to the Kafr El-Sheikh and Fayioum strains compared to the wild fish, Dr. Dorgham had to extract their DNA.
To get a good look at that DNA, the thesis uses two incredibly powerful molecular markers, RAPD and Microsatellites. Think of these techniques as a way to essentially barcode the different tilapia strains. RAPD, which stands for Random Amplified Polymorphic DNA, and microsatellites allow scientists to amplify specific regions of the fish's genome. By comparing the banding patterns that pop up from these tests, researchers can see exactly how much genetic variation exists within a single population and exactly how genetically distant one population is from another.
The ultimate goal of looking at these markers is to determine something called heterozygosity. Basically, you can think of heterozygosity as a health scorecard for the species. In any diploid organism, having two different alleles for a specific gene means there is active genetic variation. A high heterozygosity score, that means a rich, diverse, and resilient genetic pool that can adapt to changing environments and fight off diseases. A low score? Well, that means the population is dangerously inbred.
When looking at the RAPD DNA markers, the data is just incredibly revealing. The unselected, wild Abbassa population clearly contained the absolute most genetic diversity, showing nearly 45% polymorphic bands, which makes perfect sense, right? Wild populations are nature's incredibly rich biological reservoir. On the totally opposite end, the farmed Fayioum strain showed the lowest polymorphism at just 34.5%. That points directly to a severe loss of genetic diversity, highly likely due to poor breeding practices and inbreeding.
But here's where it gets really interesting. The more precise microsatellite data confirms this trend, but uncovers a completely surprising and honestly hopeful fact. Despite being a highly domesticated, selectively bred heavyweight champion fish, the Kafr El-Sheikh strain managed to maintain a high diversity score of 0.709. That is almost entirely equal to the wild fish's score of 0.711. Meanwhile, the Fayioum strain lags significantly behind at 0.652, showing a very real homozygote excess, which is a textbook sign of inbreeding. This side-by-side comparison right here is the real breakthrough of the study. It completely proves that selective breeding for larger, commercially viable food fish doesn't automatically ruin the genetics. The Kafr El-Sheikh strain is living proof that as long as commercial hatcheries manage their founder stocks intelligently, they can minimize inbreeding while still producing massive, fast-growing fish. But the Fayioum strain serves as a stark warning of what happens when that management starts to slip.
And that brings us to a really crucial realization, perfectly captured by Dr. Dorgham himself. Maintaining wild genetic reservoirs is an absolute non-negotiable necessity. The wild tilapia swimming out there in the River Nile and its canals aren't just fish, they are quite literally a genetic library. When commercial strains become too weak or too inbred, it is exactly this wild genetic variation that scientists and breeders will have to return to in order to rescue the global populations.
So I'm going to leave you with this final thought. The Nile tilapia is currently saving millions from hunger, providing this incredible, adaptable, fast-growing source of protein. But as we rely more and more heavily on science and commercial farming to feed a growing world, are we actually doing enough to protect the wild populations and the natural habitats that make all of this possible in the first place? Definitely something to chew on. Thank you so much for exploring the invisible architecture of the Nile tilapia with us today.